Hello, everyone. Good to see you. If you would, go ahead and type your first name or your nickname into the chat for me. Thank you very much. Appreciate that. Okay. So today we are going to be talking about vaccines. We're going to talk about the currently available vaccines to help us fight back against the SARS-CoV2 pandemic. Before we get to that, a couple of housekeeping things. This is officially our last meeting together. There is a meeting on the schedule for next week on Thursday at 4 o'clock, but that's more like a drop-in kind of meeting if you have any questions. So the meeting scheduled for next week at 4 o'clock is totally optional. I would ask that if you have some questions about lecture material or anything, and you want to ask them, please come to the meeting at 4 o'clock. What I'll do is I'll open the meeting up at 4 o'clock, and if no one is there, if nobody comes with questions, I'll go ahead and close it down at about 10 after 4 o'clock. So if you find that you have questions next week and you want to ask them, again, questions about the material that you've been watching in lecture or any questions about really anything, you can come at 4 o'clock and we'll talk about those. But if you find you don't have questions and you're good to go, you don't need to attend that meeting next week. So we are rapidly coming to the end of the semester. You're looking at epidemiology this week and vaccination. And next week, the final lecture topic that we do is called pathogens and how they spread. So it's essentially a sort of a final lecture about bringing everything together that we've been talking about in this unit, the epidemiology and the vaccination and the immunology, all surrounding this topic of infectious disease. And that brings us to the end of the lecture topics. Once again, if you haven't already, several of you have, thank you. If you haven't already, go ahead and type your name into the chat for me so I have a record that you were here today. And what I'm going to do is go ahead and pull up our Canvas page so we can take a look at the remaining schedule that we have. Let me get this out of our way. All right. So you should be seeing on your screen right now, the modules page on Canvas. Oops, sorry about that. That's not what I wanted to do. There we go. So I'm on Canvas and I'm on the modules page. And I'm going to scroll down here to get to this week, which is right here, the week of May 3rd. So this week, again, vaccination and epidemiology are the topics. And of course, you have quizzes associated with each one of those. And then if you look to next week, we have the final lecture topic, which is pathogens and how they spread. Our final lecture exam is then scheduled for the week of May 17th we're scheduled for May 18th. So we're going to take the exam on May 18th. It'll look just like the other exams in the sense that it's covering only the material that we covered since the last exam. So this final lecture exam is just the next lecture exam. Here was lecture exam 4 back in the week of April 19th. So since that time, we've covered immunology. This week, again, vaccination and epidemiology. And next week, pathogens and how they spread. So those will be the lecture topics that are covered on this last lecture exam. It'll be the same format of an exam, primarily multiple choice questions and also some short answer questions. It'll be about the same length as the other exams, so no surprises there. It's not cumulative. You don't have to worry about that. There will be a couple of bonus questions on this last exam. Those will be related to what we've been talking about regarding the SARS-CoV2 virus. So there could be a question about testing, diagnostic testing for the virus. There could be a question or two about the vaccines. So once again, today, go ahead and take some notes for yourself. I don't have a lot of slides for us to look at today, but some good visuals about these vaccines. So take some note, jot some notes down for yourself so you can take a try at those bonus questions on that last exam. All right, so I'm just going to pull up now. This is at the end of our syllabus. Of course, this is the tentative schedule, the last page of our tentative schedule. We are right here. We're going to be talking about the vaccines today. So as you can see, next week on Tuesday, no meeting. And then on Thursday, this meeting is totally optional. So if you decide that you'd like to come and ask a couple of questions, just come at 4 o'clock and we will talk about any and all questions at that time if anybody has them. And then here is the final lecture exam scheduled for the 18th. And that brings us to the end, which is hard to believe, but it's coming quickly. So let me ask first if anybody has any questions about lecture material or anything that you've seen on quizzes. I know you may not have gotten through all of the lecture material for the week, but if you have any questions, feel free to type them into chat and we can talk about them. My idea for today is that this is not, a typical lab for us. This is a discussion and we're going to be talking about the vaccines. So I want you to feel free to type questions out into chat as we go. I welcome you to ask questions that you may have about the vaccines. I know that there's a lot of information right now circulating on social media and on the internet and various places. And there's a lot of discussion about the vaccines right now because the vaccination program in our country, this rollout, if you will, of getting people vaccinated is certainly at its peak right now. There are lots and lots of places to get vaccinated. There are multiple types of vaccines being used. As of yesterday, I think it was announced that 50% of the people in our country who are eligible to be vaccinated, because of course, right now, you have to be over the age of 16 to get vaccinated. So half of all of the eligible people have had at least one of their vaccines. So we are rolling along, getting people vaccinated. Obviously, we're also witnessing a terrible wave of infection and death in India right now. India is literally recording in the area of 3,500 deaths per day right now from COVID. They have had millions of cases over the past couple of weeks. They are in trouble right now. Their healthcare system is collapsing right now, which is exactly what the world has been trying to prevent over the past year, is having healthcare systems collapse under the weight of this pandemic. But unfortunately, some of the decisions that the government in India has made regarding not wanting to lock down and so on has led to this terrible upswing in cases. And if you followed the news at all from India, it's just devastating. They don't have enough beds. They don't have enough hospital beds. They don't have enough oxygen to give to patients. They are working as hard as they can, but under just absolutely unbearable circumstances. So it's a good reminder to the world that the pandemic is not over. It will not be over until, I should say, unless the whole world approaches vaccination from a public health standpoint. It's not just about getting people vaccinated in the United States or, in Europe. We have to reach herd immunity around the globe, or this virus will continue to replicate inside susceptible hosts. Now that, viruses use their host to make copies of themselves. And as long as there are people who are susceptible to this virus walking around on the planet, there will be virus being replicated and spread. And of course, our other big concern right now is all of the variants, all of the mutations that occur when a virus is allowed to replicate in huge numbers, which of course this virus is still doing. Even in our country, there are still large numbers of cases and large numbers of unvaccinated people. So we are headed in the right direction in our country. Hopefully, enough people in our country will choose to get vaccinated to allow us to reach that point of herd immunity. But that's not even an absolute. In a country like ours, in a democracy like ours, people have the right to refuse vaccination, and that could be a problem for us if we can't convince at least 65, 70, 75% of eligible people to get vaccinated. We may not reach herd immunity. And if that's the case, we may be in for a difficult fall, right? We may be in for another wave of infection and disease this fall. So the pandemic is still, we're at a very vulnerable phase right now, here and around the world. You may have heard also that there's discussion right now in our government about whether or not we should waive the protections, the patent protections that we give to pharmaceutical companies surrounding vaccines. So as you can imagine, Moderna and Pfizer and Johnson & Johnson, AstraZeneca, these companies have invested a lot of time and a lot of money developing these vaccines. And it's their hope that they will reap the rewards of their discoveries. But there is a growing movement to allow for these companies, their discoveries, their vaccines, to be shared openly around the world. In other words, to allow less affluent countries to buy sort of generic versions of these vaccines. And the only way for that to happen is if companies are allowed to make generic versions of these vaccines. Right now, you couldn't do that because these vaccines are protected by patent. These companies have a patent. In other words, no one can copy their vaccine for the next so many years. I think it's 15 or 17 years. And of course, that causes a problem, right? That means, if you are a small country, a poor country, and you can't afford to buy vaccine from the United States, from the companies in the United States, what do? It's a problem. And again, it's in everybody's best interest right now that we help other countries vaccinate their citizens, that we help them get the vaccine so that they can vaccinate their citizens. But like everything else, it's a money issue. So we'll have to see what happens. We'll have to see what the World Health Organization does and what happens. I can tell you that right now there are very few vaccines available around the world. Lots of places that would like to have vaccine that don't. So, even another option might be for the United States to just continue to donate vaccine. If we don't lift the patent protections, in other words, if we're not going to allow other companies to make generic versions of these vaccines, perhaps we could donate a lot of doses around the world. Only time is going to tell, though, if that's going to happen. So let me ask first off, and go ahead and type into chat for me. What do you know about the vaccines? Just anything at all off the top of your head. Tell me some of the things you've heard about the vaccines that are available right now. Tell me what you know, or tell me things that you've heard about them. If you want to just type out how you think they work, or what they're made of, or how effective they are, or whether they have problems associated with them. You could type that into chat. Just anything that you've heard about these vaccines, the ones that are in use right now in our country. And remember, that list includes, there's a vaccine that's made by a company called Pfizer. There's another vaccine by a Massachusetts company called Moderna. There's a vaccine that was developed by the company Johnson & Johnson. And then there's a fourth vaccine that was developed by a company called Astrazeneca. So what have you heard? How many doses do you need? Is it the same for all vaccines? How many doses do you need of these vaccines? Is it the same for all of them? What have you heard about that? So Izzy's saying that she heard that the first dose of the vaccine is about 80% effective, and then the second is about 90%. There's been a lot of talk about the vaccines that need 2 doses, right? There's been a lot of talk about that. It's turned out that a good portion of Americans, at least, have received the first dose of these 2-dose vaccines, but they haven't gone back for their second dose. And when they've been polled, when they've been interviewed and asked, a lot of these people responded that they didn't go back for the second dose because they heard that you get bad side effects after the second dose. So they just don't want to go through that. So they decided to just go with 1 dose. And Morgan is saying 2 doses for Pfizer and Moderna and 1 for Johnson & Johnson. That's correct, Morgan. So for the Pfizer vaccine and the Moderna vaccine, the manufacturer recommends 2 doses for full immunization. And the Pfizer the recommendation is a 3-week interval in between the 2 doses. And Moderna recommends a 4-week interval. Now, funny thing about those intervals, they're not hard and fast. When a manufacturer for a vaccine says, get the first shot, then get the second shot 3 weeks later, the most important thing to remember about that is that you don't want to get your second dose sooner than 3 weeks, but it's okay to go a little bit past 3 weeks. In fact, you can probably go several weeks past. We have some data already to show that both of those vaccines, it's perfectly fine to wait as long as 6 weeks between doses. And that's been very helpful in places where it's just been too hard to schedule everyone so that they get the 2 doses exactly 3 weeks apart. A lot of people have gone 4 weeks or 5 weeks or 6 weeks apart, but we have some data to suggest that that's not a problem. You just don't want to go earlier than 3 weeks because it won't work as well. You need that 3-week interval. Izzy's also saying that, yes, Izzy's saying that she's been hearing some strange rumors that the government is putting tracking devices into vaccines. How many of you have heard this? Put the number 2 into chat. Just type the number 2 into chat if you've heard this theory that there is like a microchip in the vaccine, and that this is an effort by the government or by Bill Gates or whoever the deep state is. This is an effort for them to track us by literally putting a chip into us with the vaccine, the same way you might microchip your dog. Have you heard this? Yeah, Davina's saying yes. Yep, Morgan's saying yes. Yeah, this is apparently a thing. People are genuinely concerned that inside that needle, inside that vaccine, there is some kind of a tracking device. Number 1, no, there's no tracking device in the vaccine. And number 2, I hate to be the one to tell those people this, but they are already being tracked, right? Anyone who's carrying around a cell phone is already being tracked pretty much, okay? So if you're worried about the government knowing where you are, you shouldn't be carrying one of these around because they can find you with one of these. I shouldn't laugh, but gosh, I'm surprised at how many people honestly believe that these vaccines are being used as some kind of a nefarious thing, that the government is trying to do bad things to us. So Moderna and Pfizer, you need 2 shots to be fully protected. Now, in the vaccination lecture that again is this week, there's some important information about vaccines that require boosters, right? In fact, I showed you a graph in that lecture that shows antibody levels associated with first and second doses of 2-shot vaccines. If you'll give me just a sec, I'm going to pull up that graph because that's a good one for us to look at. Just bear with me for just a moment. So we've been studying vaccines long enough now in humans and in other animals that we know a lot about both just vaccines in general. Oh, sorry, actually, I'm going to pull up the immunology lecture because that's where this is, I believe. We know an awful lot about how vaccines work, and we know an awful lot about how to use vaccines in an optimal way. So I'm going to pull this graph up for us now. And again, this was in the immunology lecture, if you remember. So, oh, sorry, that's the wrong graph. No, that's the wrong graph, sorry. If you remember from the immunology lecture, we said that whenever a foreign material enters the human body, and it has to really enter, remember, it has to cross an epithelial barrier in order for the body to recognize this foreign invader. Once it does that, once it crosses an epithelial barrier and the immune system gets activated, what ends up happening is the innate immune system is the first system to encounter this foreign material. And that innate immune system, remember, is non-specific. It fights all things exactly the same way. You go through that, the symptoms of an immune response, you get fever and you get muscle aches and all the signs that you have an infection. And those are related to the innate immune system fighting off this invader. That takes a couple of weeks. That takes a couple of weeks in terms of how long it really takes for an infectious pathogen to be taken care of by our immune system. After the innate immune system is activated, it goes and activates the adaptive immune system. And remember, it's the adaptive system that is specific. The adaptive immune system is the system that stimulates B cells to make antibodies. And it's also through the adaptive system that we get immune memory. So what we really want from a vaccine is what we would get from a natural infection, which is the production of antibodies, number one, and number two, memory. We want our immune system to remember this virus, for example, so that if we get exposed to it at some point in the future, our immune system will just jump on it because it remembers that virus. So we won't get sick, or if we do, we won't get nearly as sick as we would if we weren't vaccinated. Now, I finally found the right image, so let me pull it up for us. This is the image from the vaccination lecture that shows the effects of booster injections based on antibody levels. So what you're looking at in this graph across the x-axis is the days passing by after the person gets exposed to the antigen. And of course, remember, an antigen is just something that stimulates the immune system. It's usually a protein, and it causes an immune reaction in the body. On the y-axis, we have just a general indication of the amount of antibody. So the higher up you go, the more antibody is available. So you can see that when you vaccinate somebody on day 0, for example, it's going to take a little while for antibodies to be produced. It takes a couple of weeks, as I just said. Remember, the innate system gets activated first, and then the innate system activates the adaptive system. And then it takes a full 2 weeks once the adaptive system is activated for you to get your peak level of antibody. Okay, so from the time of that initial vaccination, it takes 2 weeks. So here's day 7. Right here would be day 14. And you can see the amount of antibody is peaking right there. It takes 2 weeks for your body to produce this top peak level of antibodies against that vaccination, against that antigen. Now, here's the thing. Those antibodies do not last very long. They start to decline almost immediately, like within a week. You can see by 3 weeks, the amount of antibody has declined to about here. Now, it's not at 0, is it? If I trace this over to the y-axis, there's still some antibody, but there's not nearly what there was a week ago. So it's right around that time when the antibodies that are being produced drops down to a low level that we booster. So again, this is 3 weeks past the first shot. And for the Pfizer vaccine, that's how long they recommend, 3 weeks. For Moderna, they actually recommend 4 weeks because when they did their clinical studies, they found that the low point actually came a little bit later. It came at 28 days. Through those clinical studies, the investigators learn when the first antibodies are going to drop off. That's when you want to booster the vaccine. And as you can see, what happens after you give the booster is now you get more antibodies and the peak is going to be even higher this time. And more importantly, as that peak goes down, when it hits a low level, it's hitting a level that's still pretty high. So even after the antibodies drop down, and they will, they're going to stay at a level that's pretty good if you trace it over to the y-axis. It's pretty close to that first peak. Okay, so remember, boosters do 3 things for us. They give us more antibodies. They give us better antibodies. In other words, the antibodies bind to the virus better. And number 3, they give us a longer lasting immune response. They give us longer memory. Okay. Now, when you look at that graph, some students might say, well, wait a minute, if boosters are so important, how come the Johnson & Johnson vaccine doesn't get boosted? Sometimes for some vaccines, when they do their study and they make their graph of antibodies versus time, they notice that the booster doesn't do anything extra. For some pathogens, for some vaccines, the booster shot just doesn't achieve any additional benefit. Now, that's not saying that, they have fewer antibodies and their antibodies aren't as good and their antibodies aren't going to last. That's not what it means. It just means that for that vaccine, a booster doesn't add anything. So when they're studying vaccines, when they're making them, when they're developing them, they always try boosters in order to figure out if the booster shot is worth it, if it's worth the effort and the money. And it is if you get, again, more antibodies, better antibodies, longer lasting memory. So the Johnson & Johnson shot just doesn't get boosted, doesn't need to get boosted. That doesn't mean that people who get that shot will never need a booster. They may need a booster in a year or 2 years or 5 years. We just don't know yet. Same thing with Pfizer and Moderna. People who have been vaccinated with those may also need to be boosted, say once a year or once every 3 years or once every 5 years. We just don't know yet. Those studies are being conducted right now. They're monitoring people who have been vaccinated by measuring how many antibodies they have. They're titer. They're following their titer so that they can know when antibody production starts to fall back down again. And that will be a time that they will recommend that people get boosted. All vaccines get tested to see if boostering is important. Some vaccines require 2 boosters, some require 3 boosters. It depends on the vaccine. And then a lot of vaccines require boosters down the road. Tetanus, for example, the tetanus vaccine that you get when you're a child, that vaccine needs to be boosted about every 10 years. Even vaccines that we used to think you would never have to booster, things like, for example, the pertussis vaccine, the whooping cough vaccine that we give to babies. We used to think you never had to booster that, but we're now finding that as people live older and older lives, that it's a really good idea to booster that vaccine in our elderly people. Turns out that, as people get older and older, their ability to make antibodies against pertussis starts to go down. So in some situations, people are given boosters to that vaccine. It depends. It depends on the pathogen, and it depends on the manufacturer of the vaccine. So we have a problem in that a lot of folks are choosing not to get their second shot, their booster shot, because like we just saw on that graph, what that's going to result in is a lot of people who have antibodies that are fewer in number and don't last as long. So those people may become susceptible to the virus at an earlier date than the folks who got both shots. And that's a problem. Vaccines are developed to give us the very best that the vaccine can give us in terms of antibody production. And if you get 1 and not 2, when the manufacturer recommends 2, you're not getting fully vaccinated. So like Izzy had said earlier, when you hear things like, oh, if you get 1 shot, it's 80% effective, and if you get 2 shots, it's 90%. Those numbers are really hard to interpret when you hear them. What that's really saying is, when you get 1 shot, you're going to make antibodies. Yep. They're not going to last a whole long time, though. How long? Well, we don't know, because we haven't done all the tests yet. But if you get 2 shots, you're going to get more antibodies. You're going to get better antibodies, and they're going to last longer. You're going to get a longer immune memory. We know that. So that's why we're really trying to encourage people to get both shots. But people are funny. People don't want to experience the discomfort that sometimes comes with that second shot. So here's a question. Type into chat for me if you think you have an idea about this. A lot of people who get those 2-shot vaccines, a lot of people have been reporting that the first shot isn't too bad, but the second shot makes them feel terrible for a day or 2. So they get the first shot, not so bad. Maybe their arm's a little sore the next day. But when they get their second shot, they feel terrible the next day. They have a headache and they're really tired. Maybe even they have a fever. So why is that? Why is that? Why would you have a mild response to the first shot and a severe response to the second shot? How does that make sense? What do you think? Yep, Morgan's got it. Izzy's got it. Remember what's happening in your body from the standpoint of your immune system. This is a new virus. This is a pandemic virus. So it's new. And people around the world, unless they've already had COVID, people around the world, their immune system hasn't seen this virus yet. It hasn't encountered it. So you get your first shot, your first vaccine, you're going to mount an immune response to that. You're going to mount an immune response, sure, just like we saw in that graph. You're going to, within a couple weeks, you're going to make antibodies against that virus. Then you get your second shot. Well, now you have immune memory against that virus. So now, not only are you going to have your innate immune response, but your adaptive immune response is going to be higher. It's going to be stronger because your immune system already met this virus a few weeks ago. And so your immune system recognizes it and has a stronger reaction to it. So you, in a way, people who feel really lousy after they get that second shot, that they should feel good because they know that their immune system is working really well. Their immune system is doing exactly what it should do, which is remembering that virus and responding to it in an even more robust way. Now, I should also mention that some people don't have any bad effects after the second shot. That does not mean that their immune system isn't working very well. That's not what that means. It just means that some people have different responses. Our immune systems are all slightly different. And some people are definitely more sensitive to things like headache and muscle pain than other people are. You may know some people who, go to work when they have the flu and, you'd never know it. And, and other people are just more sensitive to the pain and discomfort that's associated with those things. So, so Morgan has a great question. Morgan's asking about this information that's been going around about the Johnson & Johnson vaccine, that it's less effective than the Moderna and the Pfizer. Poor Johnson & Johnson they've had a bit of a bumpy ride, haven't they? That vaccine had been rolled out, it had been put into use, and then a pause was put on it a couple weeks ago. They stopped using it for about 5 or 6 days because there were reports of blood clotting issues. So if you delved into that story at all, about at the time of the pause, there were 6 women, interestingly enough, who developed blood clots after getting the Johnson & Johnson vaccine. So out of an abundance of caution, the CDC recommended that we stop giving the Johnson & Johnson vaccine to people so they could study this and see if it was real, see if this blood clotting issue was the result of the vaccine or whether it was just coincidence. Remember from statistics that sometimes 2 things can be correlated. In other words, they occur together, but that does not mean that they are caused by each other. All right. So correlation is not the same thing as causation. Sometimes we see a pattern like 6 people getting blood clots after getting vaccinated, but it's not due to the vaccine. So what they spent those 6 days doing was very carefully looking at the medical history of those women to try to determine if they had any indication in their medical history that they might be susceptible to blood clotting. They tried to figure out if there was any evidence of causation between the blood clots and the vaccines. At the end of those 6 days, they had not found enough evidence to say that the vaccine caused the blood clotting. At that time, 6.5 million doses of Johnson & Johnson had been given to people, and 6 cases of blood clotting had occurred at that time. So that's less than 1 in a million. And if you remember, in our vaccine lecture, we talk about adverse effects. When it's a 1 in a million, that's what we consider a rare effect. They're often rare and severe, right? We talked about that in that lecture. This Johnson & Johnson blood clotting question would be less than 1 in a million occurrences. And so far, at least, there's no clear evidence that it was the vaccine that caused the clotting. Even if it was the vaccine, the occurrence of the clots is so low, again, less than 1 in a million, that the committee decided that the use of the vaccine provided more benefit than risk. Because, of course, that's really what we need to know. Is the benefit of getting vaccinated with that vaccine higher than the risk of developing a blood clot? And for right now, at least, the answer is yes. So Johnson & Johnson is back on the market, being used again. So that's one thing to know. Something else to know, though, about this effectiveness question, because I heard these numbers too. Remember, when the clinical studies were being done, the original reports were about the Pfizer vaccine, and it came out that Pfizer was, I want to say, 94% effective. That was the number that was sent out to the media. And then a few weeks later, Moderna released a report saying that their vaccine was 95% effective. So very, very close, very high, right? 94, 95%. Then Johnson & Johnson was released, and their number came out at like 65 or 66% effective. So a lot of people heard those numbers and concluded that the Moderna and the Pfizer vaccines were better vaccines. I mean, their protection is in the 90%, and Johnson & Johnson was only in the 60%. So of course, people assumed that those two vaccines were better. And for a while, a lot of folks didn't want to get the Johnson & Johnson vaccine because they thought it was less effective. Here's the thing that the media did not get out to people, which is too bad. The end points for those vaccines, when they were being studied, were different. For the Moderna and the Pfizer vaccines, which use the same kind of technology, the end point was, in other words, what they were trying to figure out if the vaccine prevented, the end point was hospitalization and death. So when they say that their vaccine is, let's say, 95% effective, what that means is that within a population, 95% of the people will be protected from severe disease and death. It does not mean that 95% of the people won't get COVID, does it? It just means that 95% of the people will be protected against the severe forms of COVID and death. Now, the Johnson & Johnson vaccine had a different endpoint in their study. That vaccine looked at hospitalization and severe disease and death. In other words, they used a stricter standard, a stricter endpoint. They wanted to know if their vaccine would prevent hospitalization and severe disease and death. And it came out to whatever, 66%. So you can't compare them. Statistically speaking, you cannot compare those vaccines. Moderna and Pfizer had one end point in their study. Johnson & Johnson chose a different end point in their study. So you can't compare them. But the media and the public health sort of networking system didn't really explain that to people. And so people heard two very different numbers and decided that the Johnson & Johnson vaccine wasn't very good. It's such a shame. It's hard to watch. It's hard to watch from the sidelines as misinformation and misunderstanding happens during a public health program like this. But it's bound to happen because we have not lived through a pandemic like this before. The folks that are on the planet right now, we haven't lived through a pandemic like this before. So it makes sense that we're not quite sure how to message things and how to educate people and all of that. And it's not really surprising that there's a lot of misunderstanding going around because there is. Chloe's asking a question about the other strains of the virus, the mutants, right? Or what we often hear called the variants. Remember, viruses are not technically alive, so it's technically not correct to refer to strains of the virus. That's a word that we save for bacteria, for example, because they're alive. We talk about the genus and the species and the strain of different bacteria. In viruses, we don't use that language. So instead, we talk about the variations or the variants of a virus. And that means that they have genetic differences. Their genetic material looks different. And we've heard about these, right? We heard about the UK strain, which I think is B117. And then there's a Brazil strain. There's a South Africa strain. There's now a couple of strains coming out of India. There have been lots of mutations going on to this virus because that's what happens in nature. Remember, this is a really important thing to remember about mutations in general. Every time that genetic material gets copied, there's an opportunity for mistakes to be made. So every time a cell copies its DNA molecule, for example, there's a chance that the enzyme that does the copying is going to make a mistake, put the wrong base in, and cause a mutation. The same thing happens with viruses. When cells replicate their genetic material, mistakes get made. Enzymes are not perfect. So the more that a virus circulates, the more people it infects, the more it gets copied. And the more it gets copied, the more chances that it gets mutated. So you can imagine during a pandemic, when virus is circulating around the whole world, billions of people are copying that virus in their body. Okay, so lots of opportunities for mutations to occur. And that's what's happening with this virus, with the SARS-CoV2 virus. Just like viruses always do, this one is mutating. Some of the mutations don't cause any problems because mutations, remember, are random events. They occur at random places inside that genetic material. Sometimes they cause problems, sometimes they don't. We've had a few mutations, a few variants arise that do cause problems. And the two big problems are they're easier to get and they're easier to spread. And of course, the other thing that we're monitoring for is whether or not they cause a more severe form of the disease. So there are a few variants floating around that are problematic for us as humans because they're dangerous. They're even more dangerous than the original form of the virus. Right now in our country, the UK strain, the B117 strain, is the predominant strain of virus. This mutation that it got allows it to bind much better to our cells. So the original strain, it could bind to our cells, to receptors on our cells, but this UK strain can bind much better. That means it can get into our cells much more easily. Remember, 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 as long as this virus circulates around the globe, we're going to get more and more and more mutations. It's in everybody's interest for us to get as many people immunized as possible, because the virus can't get copied inside of an immunized person, at least inside most immunized people. The virus has no place to go when more and more and more people get immunized. So that's our goal. Our goal is to get enough people immunized to get to herd immunity before more and more variants come out and get more and more dangerous with time. So you can imagine right now in India, because of how many people live in India, and because most people are not immunized in India right now, you can imagine that there's lots of virus being replicated and there's going to be mutations forming. And so public health scientists are very carefully watching the strains that are developing in India, so that if a particularly bad strain, a particularly pathogenic strain should evolve, should arise, we'll know about it when it happens. Millions and millions of people are infected in India right now. And so lots and lots of viruses circulating. It's a problem. It's a problem. We need to get people immunized quickly. We need to get as many people as possible vaccinated quickly so that we sort of beat the virus at this race. All right, I'm going to pull up, again, this little short set of slides that I have for us today, just so we can take a look at the technology that's in these vaccines, because of course it differs between the different types of vaccine. Just bear with me for just a minute. Let me get this out of the way for us. Okay, hold on just a sec. Too many buttons. Okay. So what you should see on your screen right now is a slide that says SARS-CoV2 Vaccines at the top. Now on this slide and on all the slides that I'll show you today, what you're looking at is just a little schematic, a little diagram that tries to show us what's happening with two of the vaccines that are on the market right now, the Pfizer vaccine and the Moderna vaccine. Those vaccines are called RNA vaccines. Now, what that means is that the vaccine, like all vaccines, contains some component of the virus. And in this case, the component is RNA. So in this drawing, what they're showing us, this represents a needle which has the vaccine in it. And if you can see it on your monitor, there are little blue dots in the vaccine, and they've blown up one of the dots for us here to show us what's in the vaccine. And this is essentially what's in there. It's a piece of RNA, and it is surrounded by a coating of lipid. Now, this lipid coating is the real money maker for this vaccine, and I'll explain that in just a minute. So what scientists did in the laboratory was they gathered the virus, they grew up some of the virus in cell culture. They isolated the RNA, the genetic material of this virus, and they sequenced it. They figured out the order of the bases in the RNA. They also figured out which piece of the RNA coded for the spike protein on the outside of the virus. They then made a laboratory-made piece of RNA that had that code in it, We can do that in the laboratory. We can build a synthetic RNA molecule. We can make it in any, to have any code in it that we want. So that's what they did. They built an RNA that has the same code in it as the actual virus, the code to build the spike protein. Now, when they inject this into your arm, these little pieces of RNA that are surrounded by lipid, what happens is the lipid encounters the cells that are in your arm. That's where most of us get vaccinated, up in our arm. Those little particles, the lipid around them, interacts with the lipid in your cell membrane. And those lipids just merge together. They're made out of, sorry about that, they're made out of the same material, lipid versus lipid. So they just merge together. And that allows the RNA piece to come into the cell. Now remember, RNA is RNA is RNA. It doesn't matter where it comes from. It's the exact same molecule. So inside of your cells, when an RNA appears in the cytoplasm, ribosomes are going to attach to it and they're going to translate it. They're going to build protein using the RNA as the recipe. And that's what happens. Your cells build the spike protein, the viral spike protein. And it's that spike protein that gets recognized by your immune system. Now, it's very faint in this drawing, but there's an immune cell right here that has lots of membrane extensions on it. The spike proteins that were made by your cells bind to that immune cell and set it off. This is an innate immune sentinel cell. It can bind to the spike protein and initiate an immune response. Now, this is another schematic. I liked this one. This one was actually put together by, this original one that I showed you was distributed by a hospital. They tried to, they put this on their website to try to explain to people how these RNA vaccines work. This one was on a different site, but I like this one. It shows a little bit more detail about what's happening inside of our cells. This right here represents, again, that vaccine component. The red line represents the piece of RNA, and the gray surrounding it represents that lipid. Now, they've got it listed as lipid nanoparticle coating. And again, when these particles, when they come into contact with the membrane of our cells, that's what the blue line represents. The lipid from the particle in the vaccine and the lipid in the membrane are just going to fuse with each other. That's going to let that RNA molecule go into your cell cytoplasm. And it looks just like all the other RNA. So the ribosome, which is shown in blue, is going to bind to it and start translating it. Remember, the ribosome moves down the RNA from one codon to the next, and it adds amino acids in a chain. That's what these red circles represent, individual amino acids. And once all of the RNA has been translated, you get a protein, and that's the spike protein. The spike protein that you find on the outside of the SARS-CoV2 virus. This is what we want our immune system to see, this spike protein. So by having our cells manufacture it and secrete it, the innate immune system will recognize it and the immune response will begin. Now, I said a minute ago, just a sec. I said a moment ago that the real money in this vaccine, believe it or not, is not the RNA. It's that lipid coating that's around the RNA. That is what we call the proprietary part of this vaccine. In other words, Moderna and Pfizer, they developed lipid nanoparticles that they use to coat the RNA. The code of the RNA is not a secret. That information is public. We've known from early in the pandemic exactly what the RNA looks like inside of the SARS-CoV2 virus. That was shared with all of the scientists all over the world. What Moderna and Pfizer did was put that lipid coating over it so it could go in a vaccine. Now, the reason that that is an advancement, the reason that that's such a new thing, a new technology, is because RNA is very unstable. We mentioned this in the genetics lecture earlier in the semester. RNA is very unstable. It's a single-stranded molecule normally inside of cells anyway. And it doesn't last very long. It gets degraded because it's not very stable in its chemical structure. DNA is very different. The double-stranded DNA molecule, the double helix, is very stable, isn't it? We can find DNA in at crime scenes, in blood splatter. We can find DNA on corpses that have been out in the environment, for heaven's sakes. We have extracted DNA from dinosaur bones, haven't we? DNA is a very stable molecule. RNA is a very unstable molecule. So the only way for us to have an RNA vaccine was to figure out a way to coat that RNA, keeping it safe so it didn't degrade, and allowing it to get into our cells. So the fact that they developed this lipid, that's what is so revolutionary about this vaccine. The vaccine that Pfizer has and the vaccine that Moderna has. They're both RNA vaccines. And they both figured out a way to keep that RNA safe, to prevent it from being degraded, to allow it to come into our cells so that we could then build that spike protein and generate an immune response. It really is amazing what they were able to achieve. This is also the reason, the fact that there's RNA in that vaccine, that's also the reason that that vaccine has to be kept so cold. You've probably heard that these vaccines have to be stored at -80 in a -80 freezer. And that's -80 Celsius, by the way. Very, very cold. And that's a real drawback to these vaccines in parts of the world that aren't as developed as our country is. Listen, there are lots of -80 freezers in our country. Every hospital has a -80 freezer. Every research laboratory has a -80 freezer. They're not common like a regular, refrigerator freezer would be, but there are still lots of them available to us in our country. In other parts of the world that are not as developed, those freezers are hard to come by. So the RNA vaccine has a real drawback in that you have to store it correctly or it will degrade again. It will just degrade. So that's a problem with that vaccine. Hopefully we will figure out a way to overcome that freezing requirement in later versions of the vaccine. All right, questions about the RNA vaccines. They're not complicated on paper. They're not complicated in terms of what's in them on paper. I'm sure it's very difficult to coat those RNA pieces with that very special lipid, but it really is a simple recipe. It's a piece of RNA that's been coated in lipid that gets injected into your arm to stimulate an immune response. It's an elegant vaccine. And there's no microchips in there. All right, now let's take a look at the other ones, the other vaccines. The Johnson & Johnson vaccine, which we're using right now in our country. And the other one is made by Astrazeneca. That one is not being used right now in our country, but it has applied, the company has applied to the FDA to get emergency approval. Okay. So this is a schematic that was put together by Oxford University. Oxford University worked with the company in order to, the Johnson & Johnson company, in order to build this vaccine. Okay, so Johnson & Johnson, in other words, worked together with Oxford University to build this vaccine. Okay, yeah, we don't have many more diagrams to look at. So the way that the Johnson & Johnson and the AstraZeneca vaccines were developed were as DNA vaccines. So these vaccines are also using genetic material, but they're not using RNA. Now, the first question you might have is, hold on a minute, the SARS-CoV2 virus uses RNA as its genetic material. It doesn't have any DNA in it. So how in the world can you make a vaccine against that virus using DNA? Well, what these companies did is something that we've actually been doing for quite a while, which is they took the RNA and they reverse transcribed it. Now, remember from our genetics lectures, inside living cells, the central dogma tells us that DNA gets used to make RNA in a process called transcription. Then RNA gets used to make protein in a process called translation. And of course, it's proteins that give cells their traits. Now, reverse transcription is just what it sounds like. Instead of using DNA to make an RNA, they used RNA to make a DNA. They did transcription in reverse. And the reason they were able to do that is because of an enzyme that was first discovered in the human immunodeficiency virus, in HIV. And that enzyme is called reverse transcriptase. That enzyme can use an RNA to make a DNA. So all they had to do was take the viral RNA in the laboratory, expose it to the enzyme, and they got a DNA made, a double-stranded DNA molecule. Now you might say to yourself, why would they want to do that? Why not just do what Moderna and Pfizer did and just make the vaccine with RNA in it? Well, we talked about how hard it is to work with RNA. RNA is very unstable. DNA is not. So what those companies decided to do was work with a DNA molecule instead. They knew that DNA would be more sturdy. It would be less vulnerable to breaking down and degrading. So they decided to make their vaccine using DNA. Now, the next question that they had to answer, though, was how are they going to get the DNA into our cells? They did not wrap the DNA into some magic lipid material. Instead, they used another very old method, decades old method, which is called viral vector technology. Several decades ago, in a laboratory, an adenovirus, that's what you're looking at here in blue, an adenovirus was grown up in the laboratory and it was modified. It was modified so that it could no longer cause disease. So this purple virus that you're looking at right here, that represents this viral vector. It's called the ChAd-OX1 in terms of the version that Oxford University uses. So this was originally a virus that was out in the world. It originally infected chimps. And like all adenoviruses, it caused respiratory disease. There are lots and lots of adenoviruses in the world. There are human adenoviruses too, and they tend to cause things like respiratory disease. There are adenoviruses, for example, that cause the common cold in humans. So they took this naturally occurring virus and they modified it so that it was not able to cause disease. Essentially, what they do in the laboratory with it is they take out its genetic material so that all that's left is a shell of a virus. It's got the protein capsid. It's got the spikes coming out of it because this is another spiked type of virus. But it doesn't have any genetic material in it. It's just an empty shell. And then what they did in the laboratory was they took that DNA that they had made, and that's what they're showing us in blue, and they inserted it into this shell of a virus. So now they had an adenovirus vector, a shell that contained DNA that was made from the virus's RNA. Now, why do you need the vector? Well, remember, you have to get the DNA into human cells. Even though this shell is just a shell of a virus, it still has the spike proteins. So these spike proteins can bind to our cells and allow the DNA to go inside. Now, once that vaccine gets injected into your arm, the adenovirus, the shell, binds to receptors on your cells and sends the DNA into the cells. And your cells are going to do what they do with DNA. They're going to transcribe it and they're going to translate it. So our cells will take that laboratory-made DNA. They will produce an RNA from it that would match the viral RNA. And then your cells will take that RNA and make spike protein with it. So both types of vaccines produce spike protein. And it's the spike protein that we know stimulates the best immune response. They just do it differently. One type of vaccine uses the RNA directly, and the other one uses a DNA instead. So there's one more step. That DNA has to be used to build an RNA, and then the RNA gets used to build the spike protein. The delivery method is different too. The Moderna and Pfizer vaccines deliver the RNA by covering it in lipid. That lipid can just slip into a membrane and deliver the RNA into the cell. The Johnson & Johnson and the Astrazeneca vaccines use DNA inside a viral vector, a viral shell. So the viral shell gets the DNA into our cells. And then our cells use it to build spike protein. The AstraZeneca Johnson & Johnson technology is not new. We've been doing this kind of viral vector delivery of DNA for a long time. The RNA technology is new. That lipid coating that they're using is brand new. So, what are the advantages of a DNA vaccine against SARS-CoV-2? The big one is the storage requirements. Because it's a DNA-based vaccine, it doesn't have to be kept at -80. You can store that vaccine in a regular refrigerator. So you can store that vaccine in any doctor's office, in any public health clinic, in any hospital. You can store that vaccine anywhere there's a refrigerator. That makes it a much more practical option in places around the world that are just not as developed as our country is. So it's a much more practical vaccine. The other big benefit to those DNA vaccines is that they cost a lot less to make. They don't have brand new technology in them, so they're much less expensive to make. And they're more affordable for these countries that maybe don't have as much money to spend on vaccines. So it's a really, really good thing that we have both types of vaccine available. We have RNA vaccines, we have DNA vaccines. Both of them are going to be a big, important method of getting the pandemic under control. Unfortunately, like we said, the Johnson & Johnson vaccine has already gotten a lot of bad press. So a lot of people are kind of afraid of it. Oh, I should also mention another, I forgot to say it, but another big advantage of the DNA vaccines is you only need one dose. If you think about it, I mean, that just makes that vaccine much more practical. You only have to get people to come out once. You don't have to store it at a crazy cold temperature. It's just a more practical vaccine. But right now, at least the Johnson & Johnson one has kind of a bad reputation, which isn't really a deserved reputation. We just don't have any data to suggest that that vaccine is dangerous to us. We have seen blood clots in some people, but fewer than 1 in a million. I can also tell you that the Astrazeneca vaccine, which has not gotten approval in our country yet, when that vaccine was doing its phase 1 clinical trials, its early clinical trials, there was a problem. Now, this happened back in December of 2020, and it made the news, but it didn't make a big story in the news. A report came out that they had stopped the clinical trial on that vaccine because one person in the trial, a man, I think he was in like his mid-30s, had become paralyzed after he got the vaccine. So he got this experimental vaccine, and within some short amount of time, he woke up and he was paralyzed. Now, just like they did with the Johnson & Johnson vaccine, they wanted to be very careful. So they stopped the clinical trial temporarily. And of course, they examined this man's history, his medical history, to try to determine if it was likely that the vaccine caused the paralysis or if it was something else. Now, that is very hard to do in one person. And after a long period, it was weeks and weeks, they decided that for whatever reason, and I didn't see the report, so I can't tell you exactly what they found, but they decided that the vaccine had not caused his paralysis. And they put it back into clinical trials. So the reason the Astrazeneca vaccine is not available yet is because it had its clinical trials temporarily stopped for a while. Then they got started again. So now it's ready. Now it's ready to be approved for use. And the report so far is that the United States is going to approve it. So you can look for that to happen over the next few weeks for that additional DNA vaccine to get introduced onto the market. It's all good. It's important that we have different versions of vaccines. It's important for the reasons that we've mentioned. We need to get these vaccines all over the world. We need to make sure that all countries have access to them, that all eligible people get vaccinated if possible, so that we can reach herd immunity, so that the amount of virus that's out there goes down. Remember the benefits of herd immunity. Not only are the individuals who get vaccinated protected, that's very important. Herd immunity protects the people who can't get vaccinated, the very young, the very old who might be too sick to get vaccinated, people who might have allergy problems to components of the vaccine, so they can't get vaccinated. When you get herd immunity, those people get protected because they're surrounded by people who are immune. And most importantly, during a pandemic, herd immunity reduces the amount of virus that is out there. If there are no susceptible hosts, the virus has nowhere to go. The virus has no safe haven. There are no bodies that it can get into and infect. And if it can't get into a host, it can't get replicated. And if it can't get replicated, it starts to go away. So we need to achieve herd immunity in our country, and we need to achieve herd immunity around the world if we want this pandemic to end. Questions? Questions or comments about these vaccines? Any questions about that adenovirus vector? It's pretty cool that we have that available. We use that vector for research too. We don't just use it to make vaccines. We use that vector for research projects. Anytime that we want to deliver a piece of DNA into a cell for a particular research scientific experiment, that's one of the ways we can do that, by using that adenovirus vector. It binds to the cell and it gets the DNA inside. It's one of the easiest ways to deliver DNA in the laboratory. Okay. So remember that, let's just summarize some of the things we said. Studies on vaccines determine whether or not vaccines need booster shots. Some vaccines need a booster shot because you can get more antibodies, better antibodies, longer memory. Some vaccines don't get boosted because the booster shot doesn't do those things. They study what happens when you give people a booster. And for some vaccines, it just doesn't add anything. It doesn't do those three things. You need to get the booster if the manufacturer recommends the booster. because that will give you the best protection. If you don't get the booster, your antibodies don't last as long. Your immune memory doesn't last as long as somebody who gets fully vaccinated. The effectiveness of the vaccine, the numbers that you hear, those depend on the end point that was used in the experiment. Some experiments use an easier, if you will, an easier endpoint. Some experiments use a harder endpoint. You cannot compare vaccines that were studied using different endpoints. You can't compare the numbers. So the fact that the Johnson & Johnson vaccine is reported to be 65 or whatever % effective, that's a really good number because they used a very strict endpoint. RNA vaccines have to be kept at a very cold temperature because RNA is very, very delicate and it will get degraded if it's not handled correctly. DNA vaccines don't have to be handled so carefully. RNA vaccines are more expensive. DNA vaccines are less expensive. Let's see what else. Whether or not somebody has a painful reaction to a vaccine depends on their person, their immune system. Having pain or fever or a headache, muscle aches after a vaccine just means that the immune system is doing what it's supposed to do. It's responding. That's a good thing. It's a pain for that day or those 2 days or whatever, but it doesn't last a long time. And it's an indication that the immune system is responding. And that's a really good thing. We don't know yet how long these vaccines will protect us for. That takes time. We have to follow vaccinated people over time and monitor their antibody levels. When their antibody levels start to dip down, that will tell us, okay, we need to vaccinate people again. We need to booster them again. The booster shots that are going to come down the road, the booster shots that we might all need next year or 5 years from now or whenever, the booster shots that are coming in the future, they can be modified. They can be modified to include protection against all these new variants. Just like the flu vaccine is modified every year, this SARS-CoV2 vaccine will also likely be modified every year or every so often so that we can keep up with our immunizations and keep up our protection against it. All right. Any questions? All right, everybody. All right, that's all I have for you today. So keep up with your lectures and your quizzes. Remember, next week on Thursday, I will be here at 4 o'clock. If you have any questions about the material that's going to be on that final lecture exam, come at 4 o'clock and we'll talk, okay? Otherwise, I won't see you. You don't have to come. Be sure to message me, though, if you need me for anything. Okay? All right, everybody. Have a great evening. Try to get outside if you haven't been outside. It's lovely out. And I'll talk to you soon.